Hello and welcome dear students. Today we are going to discuss about the topic genetic mapping regarding microsatellite and other DNA polymorphisms. The main objectives of today's lecture are to know the basics of genetic mapping, to know the history of microsatellites, to discuss about the mutation mechanisms and mutation rates of microsatellite mutations, to understand the biological effects of microsatellite mutations, to know the applications of microsatellites, to discuss about tandem repeat polymorphisms as well. Dear students, let's start with the introduction first. Genetic mapping describes the methods used to identify the locus of a gene and the distance between genes. Genetic mapping can describe the distance between different sites within a gene. The essence of all genome mapping is to place a collection of molecular markers onto their respective positions on the genome. Molecular markers come in all forms. So, genes can be viewed as one special type of genetic markers in the construction of genome maps and mapped the same way as any other markers. In some areas of study, the genetic mapping contributes to the creation of new recombinants within an organism. Genetic mapping offers evidence that a disease transmitted from parent to child is linked to one or more genes and provides clues about which chromosome contains the gene and precisely where the gene lies on that chromosome. Among the main goals of the Human Genome Project was to develop new, better and cheaper tools to identify new genes and to understand their functions. One of these tools is genetic mapping. Genetic mapping, also called linkage mapping, can offer firm evidence that a disease transmitted from parents to child is linked to one or more genes. Mapping also provides clues about which chromosome contains the gene and precisely where the gene lies on that chromosome. Genetic maps have been used successfully to find the gene responsible for relatively rare single gene inherited disorders such as cystic fibrosis or CFTR and Duchenne muscular dystrophy. Genetic maps are also useful in guiding scientists to the many genes that are believed to play a role in development of more common disorders such as asthma, heart disease, diabetes, cancer, psychiatric conditions, etc. There are existing dense genetic marker maps available for humans, and the introduction of next generation sequencing or NSG technologies is facilitating increased construction of genetic maps for other species as well. Genetic maps are a necessary tool for mapping of disease genes or trait loci, a method also commonly known as linkage mapping. Integrating genetic mapping and disease gene mapping with next generation sequencing has proven to be a powerful strategy in genetic research. Dear students, let's now talk about the microsatellites. A microsatellite is a tract of repetitive DNA in which certain DNA motifs ranging in length from 1 to 6 or more base pairs are repeated, typically 5 to 50 times. Microsatellites occur at thousands of locations within an organism's genome. They have a higher mutation rate than other areas of DNA, leading to high genetic diversity. Microsatellites are often referred to as short tandem repeats or STRs by forensic geneticists and in genetic genealogy, or as simple sequence repeats or SSRs by plant geneticists. Microsatellites and their longer cousins, the mini satellites, together are classified as VNTRs, that's variable number of tandem repeats. The name satellite DNA refers to the early observation that centrifugation of genomic DNA in a test tube separates a prominent layer of bulk DNA from accompanying satellite layers of repetitive DNA. They are widely used for DNA profiling in cancer diagnosis, in kinship analysis, especially paternity testing, and in forensic identification. They are also used in genetic linkage analysis to locate a gene or a mutation responsible for a given trait or disease. Microsatellites are also used in population genetics to measure levels of relatedness between subspecies, groups, and individuals. Dear students, let's now discuss the history of microsatellites. Although the first microsatellite was characterized in 1984 at the University of Leicester by Weller, Jeffries, and colleagues as a polymorphic GG80 repeat in the human myoglobin gene, the term microsatellite was introduced later in 1989 by Litt and Lute. The name satellite DNA refers to the early observation that centrifugation of genomic DNA in a test tube separates a prominent layer of bulk DNA from accompanying satellite layers of repetitive DNA. The increasing availability of DNA amplification by PCR at the beginning of the 1990s triggered a large number of studies using the amplification of microsatellites as genetic markers for forensic medicine, for paternity testing, and for positional cloning to find the gene underlying a trait or disease. Dear students, let's now discuss about the mutation mechanisms and mutation rates of microsatellite mutations. Unlike point mutations, which affect only a single nucleotide, microsatellite mutations lead to the gain or loss of an entire repeat unit and sometimes two or more repeats simultaneously. Thus, the mutation rate at microsatellite loci is expected to differ from other mutation rates such as base substitution rates. The actual cause of mutation in microsatellites is debated. One proposed cause of such length change is replication slippage, caused by mismatches between DNA strands while being replicated during meiosis. DNA polymerase, the enzyme responsible for reading DNA during replication, can slip while moving along the template strand and continue at the wrong nucleotide. DNA polymerase slippage is more likely to occur when a repetitive sequence such as CG, CG, CG is replicated. Because microsatellites consists of such repetitive sequences, DNA polymerase may make errors at a higher rate in these sequence regions. Several studies have found evidence that slippage is the cause of microsatellite mutations. Typically, slippage in each microsatellite occurs about once per 1,000 generations. Another possible cause of microsatellite mutations are point mutations where only one nucleotide is incorrectly copied during replication. A study comparing human and primate genomes found that most changes in repeat number in short microsatellites appear due to point mutations rather than slippage. Dear students, let's now discuss about the microsatellite mutation rates. Microsatellite mutation rates vary with these base positions relative to the microsatellite repeat type and base identity. Mutation rate rises significantly with repeat number peaking around 6 to 8 repeats and then decreasing again. Increased heterozygosity in a population will also increase microsatellite mutation rates, especially when there is a large length difference between alleles. This is likely due to homologous chromosomes with arms of unequal length causing instability during meiosis. Direct estimates of microsatellite mutation rate have been made in numerous organisms from insects to humans. In the desert locust, the microsatellite mutation rate was estimated at 2.1 into 10 to 4 per generation per locus. The microsatellite mutation rate in human male germ lines is 5 to 6 times higher than in female germ lines and ranges from 0 to 7 multiplied by 10 to 3 per locus per gamete per generation. Dear students, let's now discuss the biological effects of microsatellite mutations. Many microsatellites are located in non-coding DNA and are biologically silent, while others are located in regulatory or even coding DNA. Microsatellite mutations in such cases can lead to phenotypic changes and diseases. A genome-wide study estimates that microsatellite variation contributes 10 to 15% of heritable gene expression variation in humans. Dear students, let's now discuss about the effects on proteins. In mammals, 20% to 40% of proteins contain repeating sequences of amino acids encoded by short sequence repeats. Most of the short sequence repeats within protein coding portions of the genome have a repeating unit of 3 nucleotides, since that length will not cause frame shifts when mutating. Each trinucleotide repeating sequence is transcribed into a repeating series of the same amino acid. So mutations in these repeating segments can affect the physical and chemical properties of proteins, with the potential for producing gradual and predictable change in protein action. For example, length change in polyalanine tract within the HOXA13 gene are linked to hand-foot genital syndrome, a developmental disorder in humans. Length change in other triplet repeats are linked to more than 40 neurological diseases in humans. Notably, triplet expansion disease such as Fragile X syndrome and Huntington's disease are the examples. Dear students, let's now discuss about the effects within the introns, which are the coding regions of the gene. Microsatellites within introns also influence phenotype through means that are not currently understood. For example, a GAA triplet expansion in the first intron of the X25 gene appears to interfere with transcription and cause Friedreich ataxia. Tandem repeats in the first intron of the asparagine synthetase gene are linked to acute lymphoblastic leukemia. Reduced repeat lengths in the EGFR gene are linked with osteosarcomas. Dear students, let's now discuss the effect within transposons. Almost 50% of the human genome is contained in various types of transposable elements, also called as transposons or jumping genes, which were discovered by Barbara McClintock. And many of them contain repetitive DNA. It is probable that short sequence repeats in those locations are also involved in the regulation of gene expression. Dear students, let's now discuss about the applications of microsatellites. Microsatellites are used for assessing chromosomal DNA deletions in cancer diagnosis. Microsatellites are widely used for DNA profiling, also known as genetic fingerprinting of crime stains in forensics and of tissues in transplant patients. They are also widely used in kinship analysis, most commonly in paternity testing. Also, microsatellites are used for mapping locations within the genome, specifically in genetic linkage analysis to locate a gene or a mutation responsible for a given trait or disease. As a special case of mapping, they can be used for studies of gene duplication or deletion. Researchers use microsatellites in population genetics and in species conservation projects. Dear students, let's now discuss the use of microsatellites in cancer diagnosis. In tumor cells whose controls on replication are damaged, microsatellites may be gained or lost at an especially high frequency during each round of mitosis. Hence, a tumor cell line might show a different genetic fingerprint from that of the host tissue, and especially in colorectal cancer might present with loss of heterozygosity. Microsatellites have therefore been routinely used in cancer diagnosis to assess tumor progression. Dear students, let's now understand the use of microsatellites in forensic and medical fingerprinting. Microsatellite analysis became popular in the field of forensics in the 1990s. It's used for the genetic fingerprinting of individuals where it permits forensic identification, typical matching or a crime stain to a victim or perpetrator. It's also used to follow up bone marrow transplant patients. The microsatellites in use today for forensic analysis are all tetra or pentanucleotide repeats, as these give a high degree of error-free data while being short enough to survive degradation in non-ideal conditions. Even shorter repeat sequences would tend to suffer from artifacts such as PCR, stutter, and preferential amplification, while longer repeat sequences would suffer more highly from environmental degradation and would amplify less well by PCR. Another forensic consideration is that the person's medical privacy must be respected, so that forensic STRs or short tandem repeats are chosen which are non-coding, do not influence gene regulation, and are not usually trinucleotide. STRs which could be involved in triplet expansion diseases such as Huntington's disease. Dear students, let's now discuss the genetic linkage analysis using microsatellites. During the 1990s, microsatellites were the workhorse genetic markers for genome-wide scans to locate any gene responsible for a given phenotype or a disease using segregation observations across generations of a sampled pedigree. Although the rise of high throughput and cost-effective single nucleotide polymorphism, that's SNP platforms, led to the era of the SNP for genome scans, microsatellites remain highly informative measures of genomic variation for linkage and association studies. Their continued advantage lies in their greater allelic diversity than biallelic SNPs. Thus, microsatellites can differentiate alleles within an SNP-defined linkage disequilibrium block of interest. Thus, microsatellites have successfully led to discoveries of type 2 diabetes, that is TCF7L2, and prostate cancer genes, that is the 8q21 region. Dear students, let's now understand the role of microsatellites in population genetics. Microsatellites were popularized in population genetics during the 1990s because a PCR became ubiquitous in laboratories. Researchers were able to design primers and amplify sets of microsatellites at low cost. Their uses are wide-ranging. A microsatellite with a neutral evolutionary history makes it applicable for measuring or inferring bottlenecks, local adaptation, the allelic fixation index, that is FST, population size, and gene flow. As next generation sequencing becomes more affordable, the use of microsatellites has decreased. However, they remain a crucial tool in the field. Dear students, let's now discuss the limitations of microsatellites. Repetitive DNA is not easily analyzed by next generation DNA sequencing methods which struggle with homopolymeric tracts. Therefore, microsatellites are normally analyzed by conventional PCR amplification and amplicon size determination. The use of PCR means that microsatellite length analysis is prone to PCR limitations like any other PCR amplified DNA locus. A particular concern is the occurrence of null alleles. Occasionally, with a sample of individuals, such as in paternity testing casework, A mutation in the DNA flanking the microsatellites can prevent the PCR primer from binding and producing an amplicon, creating a null allele in a gel assay. Thus, only one allele is amplified from the non-mutated sister chromosome, and the individual may then falsely appear to be homozygous. This can cause confusion in paternity casework. It may then be necessary to amplify the microsatellite using a different set of primers. Null alleles are caused especially by mutations at the 3' section where extension commences. Dear students, let's now discuss the other polymorphisms at DNA level, that is tandem repeat polymorphisms. The term tandem repeats are used to describe tandemly repeated DNA sequences, also known as simple sequence repeats or SSR, or satellite DNA, which includes both microsatellites and mini-satellites. Tandem repeats can involve mononucleotide, dinucleotide, trinucleotide, that is triplets or tetranucleotides, etc. Microsatellites range from 1 to 5 to 1 to 10 base pairs in length. Thus, tandem repeats with a motif longer than the upper limit for microsatellites, that is greater than 10 base pairs in length, are generally called mini-satellites. So, tandem repeat polymorphisms or SSR polymorphisms have also been referred to as variable number tandem repeats or repeat length polymorphisms. DNA sequences that are repeated in tandem are common throughout the genomes of a wide range of species, including humans, and are often roughly conserved, suggesting that they have an important function. Most research on tandem repeats has focused on those in the human genome, such as microsatellites used as genetic markers and as repeat expansion mutations causing dominant or recessive disorders with Mendelian inheritance patterns. The term dynamic mutation has been used to describe the variation in tandem repeat length that has been found in many human genes to cause a range of repeat expansion disorders, particularly those affecting the nervous system, including Huntington's and other polyglutamine diseases, Friedreich's ataxia, and Fragile X syndrome. It should be noted that one of the genes, that is FMR1, which contains a trinucleotide expansion implicated in Fragile X syndrome, shows further associations of permutation repeat lengths below the disease threshold necessary for Fragile X syndrome, with more complex disorders including tremor, ataxia, Parkinsonism, neuropsychiatric symptoms, and premature ovarian failure. A much larger array of tandem repeats is present in and between genes that are not known to be involved directly in disease of Mendelian inheritance. Tandem repeats are also referred to as simple sequence repeats, microsatellites, or mini-satellites. These repetitive sequences can be located in exons, introns, or intergenic regions, providing opportunities for the modulation of gene expression and for that of the structure and function of RNAs and proteins. For example, codon repeats translated into amino acid runs. Dear students, let's now discuss about the short tandem repeats. Short tandem repeats are very short stretches of DNA that are repeated back to back at various locations throughout the human genome. Typically, the repeating sequence is just 2, 3, or 4 base pairs in length, and the number of copies found back to back is variable across a wide range. Unlike the case for the DNA sequences of coding genes, there is no correct number of repeats for any specific short tandem repeat or STR in the genome. They are simply areas within the genome in which variation is normal and healthy. For any specific STR, each person will have 2 copies, one that is inherited from his/her mother at conception and the other that is inherited from his/her father. STRs are helpful in forensics and paternity testing because there are a lot of natural variations in short tandem repeats. the chance of two people matching for the exact number of repeats on both inherited copies of the STR is fairly small. On combining analysis of many STRs across the genome, the probability of two people matching by a random chance was found to be extremely low. The use of Y chromosome STR loci has become increasingly common in forensic science, partly because of the loci being recombination free during meiosis and paternally inherited. The analysis of Y chromosome STRs in paternity testing of potential paternal relationships is very important, especially in cases in which a potential father is not available. Dear students, let's now discuss about the copy number polymorphism or variation. Genetic association studies generally evaluate SNPs, which are variations in single nucleotides at a specific genomic location between individuals of the same species. Recent studies indicate that the human genome contains another frequent type of polymorphism, copy number variations. A copy number variation or CNV is a variation in which a segment of DNA can be found in various copy numbers in the genomes of different individuals. CNVs or copy number variation range in size from a few hundred nucleotides to several megabases. Compared with SNPs, CNVs affect a more significant fraction of the genome and arise more frequently. Hence, CNVs significantly contribute to human evolution, genetic diversity, and an increasing number of phenotypic traits. CNVs of DNA sequences are abundant in natural populations and are functionally significant but still need to be fully ascertained. A CNV or copy number variation is generated by both recombination and replication mechanisms and a de novo locus specific mutation rate, which is higher than in SNP. CNVs can cause Mendelian, sporadic, or diseased effects and affect gene duplication, exon shuffling, and genome diversity and evolution. Dear students, let's now discuss about the single nucleotide polymorphism. A single nucleotide polymorphism is present at a particular nucleotide site. The DNA molecules in the population often differ in the identity of the nucleotide pair that occupies the site. For example, some DNA molecules in the same population may have a TA base pair at a particular nucleotide site, whereas other DNA molecules in the same population may have a CG base pair at the same site. This difference constitutes an SNP. The SNP defines two alleles for which there could be three genotypes among individuals in the population. Homozygous chromosomes or heterozygous chromosomes with TA in one chromosome and CG in the homologous chromosome. About 3 million single nucleotide polymorphisms that are relatively common in the human population have been identified, of which about 1 million are typically used in a search for SNPs that might be associated with complex diseases such as diabetes or high blood pressure. In some instances, a variant form of a gene may confer an evolutionary advantage to the species and is eventually incorporated into the DNA for many or most members of the species, and effects of the variant form may be both beneficial and detrimental depending on the circumstances. For example, a heterozygous sickle cell mutation confers resistance to malaria, but a homozygous sickle cell mutation is usually lethal. Dear students, let's now conclude the lecture on genetic mapping. Genetic mapping refers to the techniques used to identify a gene's location and distance between genes. The distance between various sites inside a gene can also be described through genetic mapping. Placing several molecular markers at specific locations on the genome is the fundamental element of all genome mapping. There are many types of molecular markers. When creating genome maps, genes can be observed as a particular class of genetic markers mapped similarly to other markers. Genetic polymorphism is the inheritance of a trait controlled by a single genetic locus with two alleles, in which the least common allele has a frequency of about 1% or greater. Sources include single nucleotide polymorphisms, sequence repeats, insertions, deletions, and recombination. Changes in DNA sequence that have been confirmed to be caused by external agents are also generally called mutations rather than polymorphisms. So, genetic mapping can offer firm evidence that a disease transmitted from parents to child is linked to one or more genes. Mapping also provides clues about which chromosome contains the gene and precisely where the gene lies on that particular chromosome. Dear students, it was all about today's lecture regarding genetic mapping like microsatellite and other DNA polymorphisms. Hope you have understood well. See you next time with a new topic. Till then, take care and goodbye.